My pleasure to welcome our next speaker, Yogev Debbi from Mina Bio.
Hi, everyone. So, Yogev Debbi, co-founder and CEO at Mina Bio, and we unlock gene therapy with our non-viral delivery system, AI platform for lipid nanoparticles. So thanks, Corey, for giving some background. So we are a team of repeat founders with exits across AI, software, and chemistry, working for the past 20 plus years together, backed by top biotech investors, A16Z, NFX, and others. We are blessed to be surrounded by a team of experts in our advisory board, and Corey just presented, so shout out for you, Peter. Also, I saw him in the crowd. And this slide, I actually wanted to start with that. That was made by Chardan Investment Bank, actually by Kip that is here in the crowd. I didn't know he's joining us. So what you see here is the genetic medicine indices actually outperform all the other indexes from the beginning of the year. So there is some room for optimism, right? I mean, we're here for patient impact, but also capital is important, and we have some room for optimism at the beginning of this year. So delivery is hard, we all know that. And the main reason is because we don't know the underlying biology. We don't know enough about the fundamentals. So biology is not engineering, and this is why things take tremendous amount of time and money until they work. And most things don't work. So at Mina, we have actually closed the loop between AI and lab. So we have leveraged this engineering cycle of design, build, test, learn cycle, which make us produce hundreds of data points every week. So we design candidates by our AI. We synthesize lipids, we make the formulations in our lab, and then the important part is learn, where all the experiment results go back to the database to make it smarter and better. So the two main data sources that we leverage, one is from our literature, one is from the literature, and the other one is from our own lab. On the literature, we actually collected all the public data, so patents, publications, research papers, and we formatted, curated, and tagged all the data out there in our database. You can see here a few examples of how you scrape the data. In our lab, we not only leveraging high throughput data generation, we also build software tools that go and make sure that all the protocols are being followed, the data is QC'd, and eventually uploaded to the database, to the training set, to add data points. In fact, by now, we probably have the largest LNP, the public data LNP that is out there. Probably every LNP that ever published is in our database. And so one thing we did in order to accelerate science in the community, we took all of this data and we built the first of its kind LLM-based AI agent for LNPs. So if you want ChatGPT for LNP, you're welcome to try it. This is on our website. It's free. And this is something that we contributed, and we'll talk about data later on. The first point that I wanted to present is how we share data in the community and how we leverage that for all of us. We'll talk about it later. So a few examples of how the technology actually works. So, okay. So this is a high-level architecture of how we actually work. So you have data in, then several layers of data digestion, and on top of that, we build machine learning models that actually predict and optimize for different characteristics, such as physiochemical characteristics like size, data potential, electric charge, etc., activity in different cell types, safety, manufacturability, etc. So we have dozens of models that actually go and optimize for, you take the combination of different models, and this is how you get highly customized LNPs for different targets, having it specific targets for specific tissues or other areas. Using this approach, we have shown over and over again, and I'll try to quickly by the data, that in this tech, leveraging tech and AI, you can actually be much more capital efficiency than traditional biotech, and hence leverage much less animals, like 10 times less animals, cost, and time in the process. Okay, I'll skip that. Okay, wow. So first example, this is an example of a lung-specific LNP, okay, which is systemic administered intravenously. And here we were able, what's the next slide? Okay. So here we were able to actually build an LNP which, as you can see, high expression in lung, high specificity, nothing gets expressed in liver. We did it in 6 months of development, and it got translated into primates. So that's point number one. Another example is T cells. So Corey mentioned the CAR-T. So this is a passive targeted LNP, which means we didn't add any targeting moiety, so no antibody or anything like that. Here in 4 months of development, we were able to go from generating data, building models, and get into nice primate validated data of transfected T cells. And there is no liver, also we didn't have the chart here, but no liver expression at all. Another kind of unique program that we have developed is optimization for safety. So this is really important in today's, all the immunogenicity and toxicity, all of that. So we built a safety optimization model for LNPs. And what you can see here, we took the lung LNP, and after 6 months of development, we generated data over different cytokines, liver enzymes, coagulation, and others. We built models for each and every one of those. And then we got an LNP which was more safe. So we doubled the therapeutic index, and it was validated in primates. I don't have the data here. It was validated in primates, and this is comparison to benchmark, which is SM-102 is the Moderna COVID vaccine, so just in terms of safety. And this is actually a unique thing. So usually when talking about LNPs, you have lung data, T cells data, you have liver data. For AAVs, you have brain data, you have muscle data, but this is pretty unique. So here, that shows the strengths of leveraging. Sometimes it's text, sometimes it's a high throughput screening like what Corey does and did in the past. But this is really, really interesting. So intravenously injected of LNPs, which got expression of mRNA in all the cartilage tissues, so all the joints of mice in this case. So pretty unique. I knew you loved it. You love it. So at Mina, we have a wide selection of delivery assets that already validated in Vivo over the course of 4 years that we exist. Okay, let's go over. We're a team of 20 people, really diverse team between data science, Gen AI, chemistry, biology. We're based between Tel Aviv and Boston. The business development is in Boston. The rest are in Tel Aviv. And that's it. Thank you so much.